Hello everyone, my name is Shriya Kohli and my topic for today's presentation is translation. So, what is meant by translation? Many of us have seen movies in a language we don't understand. So, how do we watch them? Either we listen to the dub version or we read subtitles, which are nothing but the translation in our own language. So, basically, translation is conversion of words or texts from one language to another language which can be easily perceived by us. Moving on to the definition of translation, it is a process by which proteins is formed from mRNA. Here, the language of nucleotide sequence on mRNA is translated into the amino acid sequence for protein synthesis. And this occurs in cytoplasm and requires many things like amino acids, tRNA, mRNA, ribosomes, energy sources like ATP or GTP, and protein factors. Moving on to the step of translation. There are basically four major steps in translation. First is the charging of tRNA, second is initiation, third is elongation, and fourth is termination. So, talking about charging of tRNA. We know that a tRNA has an acceptor arm and this acceptor arm binds with a particular amino acid. So this process of attachment of amino acid to the acceptor arm of tRNA is called charging of tRNA. In the particular diagram, we can see that tRNA binds to a particular amino acid and forms amino acyl tRNA with the help of an enzyme, amino acyl tRNA synthetase. It is a very crucial enzyme which helps in this process. So this charging of tRNA is a high energy requiring process. It needs two high energy phosphate, or we can say two ATP equivalents. The DHU arm of the tRNA recognizes this enzyme, this amino acyl tRNA synthetase, and this is when the specific amino acid joins with the acceptor arm of tRNA. Now moving on to the next step, that is initiation. Here in the diagram, we can see that there is a polypeptide chain shown in blue shade and an mRNA chain shown in red. So let's say if this is a polypeptide, we know that the first amino acid that will be synthesized will be methionine. And methionine is coded by only one codon, that is AUG. So AUG will be present in mRNA chain. Now here in this mRNA chain, we can see that there are many AUGs we can see. So how to recognize which one is the start codon? To recognize the start codon, prior to the first AUG, there is a marker sequence highlighted in green color. The first AUG that comes after the marker sequence is called the start codon. And this marker sequence is known by different names. In prokaryotes, it is known as Shine-Delgado sequence, and in eukaryotes, it is known as Kazakh sequence. Initiation is helped by some initiation factors, as mentioned, IF in prokaryotes and EIF, eukaryotic initiation factor in eukaryotes. Moving on to the steps of initiation. There are four basic steps. First is the disassembly of ribosome unit. Second is the formation of 43S pre-initiation complex. Third is the formation of 48S initiation complex. And the last step is the formation of 80S initiation complex. Moving on to the first one, disassembly of ribosomal unit. In the particular diagram, we can see that the ribosomal units 40S and 60S move apart from each other. This is initiated by two initiation factors, that is EIF3 and EIF1A. These two initiation factors keep these two ribosomal units separated. Moving on to the next step is the formation of 43S pre-initiation complex. From the flowchart, we can very well perceive what exactly happens in this step. So to begin with, GTP binds with an initiation factor, EIF2, and forms a binary complex. Now to this binary complex, an initiator tRNA, which carries methionine as an amino acid, binds, which leads to the formation of a ternary complex. In the ternary complex, we can see GTP, EIF2 and initiator tRNA. Now to this ternary complex, 40S subunit binds and leads to the formation of 43S pre-initiation complex. We can very well see in the diagram, 43S pre-initiation complex consists of 40S subunit, GTP, EIF2 and initiator tRNA. The next step is formation of 48S initiation complex. In this step, the mRNA binds to the 43S pre-initiation complex and leads to the formation of 48S initiation complex. The last step is formation of 80S initiation complex. In this step, the 60S subunit binds to the 48S initiation complex. The difference is that the GTP is hydrolyzed from the 40S subunit and it moves away. So, the 80S initiation complex consists of 60S subunit, 40S subunit, EIF2 and the initiator tRNA along with the mRNA chain. Now we are going to see the sites in 80S initiation complex. There are certain sites in the 80S initiation complex. These are P site, A site and E site. Now it is very important to know that where exactly the initiator tRNA is located. The initiator tRNA is located in P site and the A site and E site are vacant, they are free. So we completed the initiation step. Now moving on towards the elongation step. Elongation is helped by certain elongation factors. It is a multi-step process and it consists of 3 basic steps. First is the binding of new amino acyl tRNA to A site. Second is the peptide bond formation. And third is the translocation of ribosome on mRNA. Starting with the first step, that is binding of new amino acyl tRNA to A site. In this particular diagram, we can see that the initiator tRNA is located in the P site. Now, let's say this A site is having codon UUU. We are very much familiar with triple U. What does UUU codes for? Yes, it codes for phenylalanine. So something is happening in the cytoplasm related to phenylalanine. What exactly happens in cytoplasm is the charging of tRNA occurs. As I said, charging is a process in which a particular amino acid binds to the acceptor arm of tRNA. Since A site consists of UUU, in cytoplasm, phenylalanine binds to the acceptor arm of tRNA and this new tRNA which carries phenylalanine binds to this A site. This process of binding of new tRNA to A site is helped by elongation factor 1 and hydrolysis of GTP. So, with the help of these two things, charged tRNA binds to the A site. Once the binding is done, we can see the net result as the A site consists of new tRNA carrying phenylalanine, P site consists of the initiator tRNA with methionine and the E site is free. So, let's move on towards the next step. Next step is the formation of peptide bond. In peptide bond synthesis, what happens is the amino acid which is seen in the P site will form the peptide bond with the amino acid in the A site. Methionine is seen in the P site as we have already discussed. So the carboxylic group in the methionine will form a bond with the amino group of phenylalanine and this is what referred as peptide bond formation. This process requires peptidyl transferase activity and very importantly, no energy is required in this process. So what is the net result of peptide bond formation? We can see here that the A site consists of a polypeptide, the initiator tRNA with the phenylalanine. The P site consists of the exiting tRNA as the methionine has already formed a bond with phenylalanine in the A site and the E site is free. Moving on to the next step is the translocation of ribosomes on mRNA. This is a very important step. So, what exactly happens in this step is that the ribosomes are moving on the mRNA. Just assume that the mRNA is static and ribosomes, the 40S and 60S, are moving one codon length in the following direction as shown in the arrows. So, what is the net result of translocation? See very carefully. Now, the result is that the P site now contains the polypeptide chain. In the previous site, we can very well see that the A site was containing the polypeptide chain, but because of translocation, now this polypeptide chain moves to P site and the exiting tRNA. The exiting tRNA in the previous slide, we can see it was in P site, but now due to translocation, the exiting tRNA moves to E site. So what is the net result? The net result is that the P site consists of polypeptide, initiator tRNA with phenylalanine, A site is left free, and the E site consists of exiting tRNA. Now, let us say that this A site is now vacant for AGA. Now we know that AGA codes for arginine. So again, in the cytoplasm, there will be charging of tRNA. Arginine, the amino acid, will bind to the acceptor arm of tRNA. This new tRNA will bind to A site. Again, peptide bond formation will take place between P and A site. Again, translocation will occur. The polypeptide will shift to P site. A site will again be left vacant. Again, a new tRNA will come, bind here. Translocation will take place. Peptide bond will formation take place. This process will go on and will lead to formation of a polypeptide chain. So, now moving on to the next final step, that is termination. Termination is specified by stop codons in A site. Look at this particular diagram. When a particular stop codon like UGA or UAA or UAG come at A site, it is a signal that now it is the time to stop translation. When the stop signal or when the stop codons comes in A site, then the A site is bound by certain factors, which is called releasing factor. These are releasing factor 1, releasing factor 3 and GTP. So when these releasing factors attach at A site, the polypeptide chain which is formed at P site because of translocation and the previous processes, this polypeptide chain is freed. It is liberated from this translational machinery. As you can see in this particular diagram, the A site, the releasing factors attach to A site and the polypeptide chain is freed. It moves away from the ribosomal machinery. Further, what happens now is that this translational machinery also disintegrates. Now the 40S subunit also disintegrates, 60S also is separated and the mRNA chain is also separated. So this is termination, the final step of translation. This process needs peptidyl transferase activity. So that was all about translation. I hope you guys liked it. Thank you.